Well, I want to start by thanking the organizers for allowing me to bring this work to you. I'm very excited to do so. And I have nothing to disclose. So I think you guys have seen a few of these funnel diagrams over the course of this meeting. But I just want to reiterate that we've gotten pretty good at identifying variants in clinical samples, but we still have a major bottleneck at this point of variant interpretation. And part of this really does stem from vast literature that is out there and how quickly that is continuing to grow. But part of that is also, as Alex pointed out in the previous talk, that some of these clinical interpretations are often being curated into private academic silos and restricted access commercial databases. And a lot of times these interpretations have very limited provenance and no real mechanism for feedback. And to address some of these issues, we thought a public open domain effort would be the way to handle this. And so with those things in mind, we created CIViC. And CIViC is an open access, crowdsourced knowledge base and curation system for clinical interpretation of variants in cancer. And so we're talking about variants that have significance in cancer. What are we talking about when we're in the context of CIViC? And so we primarily focused on 4 areas: those variants that are prognostic of survival change, predictive of therapeutic response, diagnostic of tumor subtype or tumor type or even exclusive of a diagnosis, and those that are predisposing for cancer development. And while most of the variants that are, or a lot of the variants that are in CIViC are really focused on somatic events, we do include predisposing germline events as well. So one of the big goals of CIViC is really to take this sort of information from the peer-reviewed public literature. I do want to reiterate that this is not patient observations. This is the existing medical literature being curated. And so taking information like this and turning it into something like this, which is both human readable but very structured. So we have structured fields that are linked to ontologies, as well as making this human readable. And so these structured fields include things like we assign an evidence level. So this could be validated information, such as things in WHO guidelines or NCC guidelines, information that is from clinical trials, case studies, and we also support preclinical work. And then we also assign an evidence type, much as I had just described: prognostic, diagnostic, et cetera. And then we assign a clinical significance. So if it's, say, diagnostic, it's a positive association with a diagnosis for that disease or subtype of disease, or a negative association, because sometimes variants can be exclusive of a disorder as well. And very quickly, you can look at this evidence item, and without reading the human readable statement, you can gather that this is prognostic of a poor outcome in papillary thyroid cancer. Additionally, as I said, it's linked back to the original source. So if you have any questions, if you want more information, you can very quickly go back to that original source. Additionally, a single source can produce multiple pieces of evidence. So we're separating out all the different pieces of evidence from a single publication. So a single paper could talk about prognostic items and predictive items, but also multiple variants, multiple genes. This happens all the time, right? So this is actually separating out these pieces. So a single resource can provide multiple evidence statements that are linked to, and a single piece of evidence is linked to a single variant and a single gene, although genes can have multiple variants associated with them, and variants can have multiple pieces of evidence associated with them. But again, this evidence statement is just looking at one piece from this paper, and then we also assign an obviously subjective rating, but we do give a trust rating for that piece of evidence, because on a whole, a paper could be focused on a few variants that they went and did a lot of follow-up data on, but there will also be other variants and other interesting results that you do want to capture, but may have not been the main thrust of the paper. So maybe there's a little less evidence backing that up, so you want to give it a lower trust rating. Additionally, with this evidence, you can see the full history of this evidence. Across the top, you can see who submitted it, who it was last modified by, and all of those sorts of things. If you hover over that, it actually gives you a timestamp as to when that happened. But on this Evidence Talk tab, you can actually see the full history, including very detailed diffs. So you can see every single character that was changed. But then you can also see the comment history. So if there was any discussion or confusion related to the evidence item, all of that is captured there. And say you were an author of a paper and you disagreed with our interpretation, as Dr. Van Allen pointed out very nicely the other day, you can suggest a change. So you simply click on the pencil icon and you can suggest changes to the interpretation of this piece of evidence. And so you can make these changes at the evidence level, which I'm showing you here, at the variant level, which also has curated aspects and structured data, or at the gene level. And as I said, and this is open source, crowdsourced, and so anybody can make these changes. However, this is not just blanket, anybody can change anything at any time. All of the changes that are made are actually moderated by expert editors. So right now, if you have your laptop open or anything else, you can go create an account, and then you are a curator in CIViC. Congratulations. So, and once you've done that, you can now submit evidence to CIViC. And so we do that for provenance so that you can get credit, but we can also see the history of what has been undertaken on a particular piece of evidence or any changes. But it doesn't cost anything, and you don't really even have to give your email address if you don't want to. So please do so if you have an interest. But then your submitted evidence is evaluated by an editor. That editor can then accept that evidence and say, yeah, we're good to go, and accept that into a moderated state, or reject it if there are gross issues with it. If you really aren't following the CIViC model and it's not really recoverable in its current state, then on rare occasions we do reject evidence. But most of the time, you end up in this kind of like editorial cycle where you suggest a change and then it goes through another round of review, and then you get into a nice space and then have it accepted. But the important thing is evidence is visible in all steps of this process. So even if a moderator hasn't gotten there, you can still search for it, you can still find it, and still use it as a useful resource to yourself. And maybe you want to take it with a little bit more hesitation because it hasn't been moderated, but you can still go look at that paper for yourself and use it. So I don't have a ton of time today, so I'm going to this is just a nice screenshot of our website, and you can visit it at civicdb.org. But what I really came to talk to you about today is actually assertions. And so assertions are another portion of the CIViC knowledge base that aggregates multiple CIViC evidence items into a single clinical consensus. So this is focused on a specific variant, a specific disease, and a specific evidence type. So this is more along the lines of the sort of information that you'd see in ClinVar. And in fact, I'm very happy to report that we've made our first submission from the CIViC interface to ClinVar. And in this method, the underlying data is very carefully defined, which allows you to rapidly reassess it. So we've got an assertion in a happy state where, while there's some more papers that have been published, all of that is in one place. We've already evaluated it, and it really kind of speeds up the process of thinking about this again. And so this is what an assertion looks like. And there's a single one-sentence summary that says what this assertion is about, what the clinical significance of this variant is in this context. And underneath of it, you have all of the pieces of evidence that went into that. You can actually click that little evidence cards tab and see each of those evidence items in detail and just scroll very quickly through that. But all of them are there, and you can see there's a range of evidence there from 1 stars to 5 stars, and actually you can't see all of them. There are 14 evidence items here, but you can see the first 7, and you can see that there are validated evidence items, there are clinical ones, and there are also some case studies mixed in here. And this is a place that we can actually, we can assign a lot of the new guidelines that have been released. So here's where we can add our AMP categories, or a tier and level. This is also where we can annotate things like this is in the NCCN guidelines. And we can also annotate information like this has been through regulatory approval, or we know that there's an FDA companion test that exists. So just as I walked through the cycle of that evidence goes through, assertions do the exact same thing. So it's always visible, it's all there, so it's also useful in that regard. But the main thrust of this talk is really actually like, how do we get assertions to an accepted, editor-approved state? So we've come up with a few minimum guidelines for getting it to this state. And so these are some of the rules for accepted assertions. So that doesn't preclude you from submitting an assertion that isn't quite in its finalized state, but this is what would have to happen for it to get to an accepted, moderated, approved state. So the gene and a variant must exist in CIViC. There has to be evidence in CIViC supporting this this assertion. That's pretty self-evident. But then the evidence must be fully curated. So we actually, as moderators, need to go through and make sure all of those evidence items are in a finalized sort of moderated state. That doesn't mean they can't further be edited, but when we're evaluating the assertion, those need to be in a nice, neat place. But then they can always be additionally changed and improved. So we also have this description section, which is personally my favorite, because the one-sentence description doesn't tell me, it tells me overall what's going on, but you lose so much of the details. And so the description is really about explaining any critical rationale. Are there evidence items that disagree with each other? You need to explain why you support one way over another. If you want to bring in additional information that maybe isn't captured by our structured fields, this is a great place to do that. Maybe a nomad allele frequency. We do show them here when they're available, but maybe you want to expound on that a little bit. So evidence items that support an accepted assertion, you need to have at least one with 3 or more stars. So I mentioned these trust ratings. And if you look at, and I'm not saying all of them have to be that, as you saw, we had a range from 5 to 1 stars there, but we want them to at least have some evidence of average quality or better. And you can actually see from over 3,000 evidence items that have been accepted in CIViC, we have really, the criteria that we use are actually working pretty well. So from 1 star to 5 stars, 5 stars being the best, our 2 outliers are, we've got only about 5% of the database has either 1 star or 5 stars, so the best and the worst. And then we have 2 and 4 stars, about 25% of the database each. And then the vast majority of things in there are average evidence, which is exactly what you'd expect. So 3 stars for us is evidence is convincing but not supported by the breadth of experiments. Maybe it's smaller scale projects, but any discrepancies that might be described are actually explained and not really concerning. So you want that or better to be associated with your assertion. So how does this fit in with the AMP-ASCO-CAP guidelines that have been released? So for this one, we had a tier 1 level A evidence item, assertion. And our criteria for that, for either tier 1 level A or tier 1 level B, is at least 1 CIViC level A or B evidence item is there. Now you can see here we have multiples that fulfill that criteria, but this is the absolute minimum, because there are diseases that are super rare or variants that are super rare that might not have as high a bar as in this case lung cancer and EGFR mutations. So that's how this fits into, I guess, our AMP-ASCO-CAP tier and level. And I know it's confusing with multiple levels and multiple organizations, but hopefully this helps illustrate this a little better. But tier 1 level A or level B is validated, needs to have evidence items that are either validated, or at least 1 evidence item that is validated or clinical. For tier 2 level C evidence, according to AMP guidelines, we want at least 1 of those level A or B evidence items, or 2 or more level C evidence items. And for the tier 2 level D evidence, we want at least 2 case study style evidence or preclinical studies that support this assertion. So in summary, we require that there's a gene variant must already exist in CIViC. Underlying evidence is fully curated. At least 1 evidence item has 3 or more stars. You have a concise, clear summary statement that's not confusing. And that your description explains any discrepancies or nuances that are needed here. And we really recommend that this is the current state of the field sort of summary, that the evidence under there actually is an aggregate of multiple studies and where the state of the field is. And they don't have to match, as Alex very pointedly pointed out, is that sometimes the evidence isn't the exact same variant of the exact same amino acid. You have related things that should be associated with this assertion. We also recommend that you use Pubmed Sources for the most part. We do support ASCO abstracts, and we can already think of reasons why you'd want to create an assertion that's based on ASCO abstracts, particularly prematurely discontinued trials that never make it to publication. The negative results are just as important as the positive results sometimes. So very briefly, because I have almost no time, CIVic's really enabling international collaboration. And this was an in-person meeting that we had in 2018, and just showing the origins of people that were using it and that came together for this. And I know there's a lot of organizations that have expert working groups, and they're working on widely different schedules. So having a resource like this to come together and work on different schedules is great. So I will go ahead and I'll skip my conclusions. I'm over time, and I realize that. But I do want to really quickly thank the CIViC community, because without them, this could not be possible. Members of the McDonald Genome Institute, and particularly the Griffith Lab, you've actually got to hear quite a bit from them at this meeting, and they're fabulous to work with. And then, of course, our funding agency, the NCI.